Did anybody see the seminar at all? It was pretty cool. I should have assigned it or something. It was the basic gist of it. It was a professor at UCSF in the radiology department, and they do a lot of cool stuff, like almost like a combination of organic chemistry and radio imaging, like PET scans and things like that. A lot of cool organic chemistry, but in the end, they're basically imaging tumors and various kind of like protein misregulation and things like that by using radio tracers, and like a molecule labeled with a like an antibody labeled with a radioisotope that they can detect on very small concentrations. I have a friend that works in the radiology department there. One of the things that they do that's pretty wild is they do organic synthesis, incorporating like a radioactive fluorine or something like that into a molecule. And then the half-life will be a few hours. So they incorporate it and then they inject it into the patient immediately, and they're able to do the imaging experiment kind of like within an hour, half an hour, or something like that, and actually get like real clinically useful data. I have a little bit about radio, well, we'll say in vivo imaging, which is the general technique, that I'm going to present a little bit later in the course. I have a couple cool slides that I have kind of came up with. All right. Okay, so let's see. What first? I'm a little slow to grade, as you might notice, because I'm teaching my organic 1 class. And anyway, so I'll try to keep up with the grading, but right now I am trying to get a couple good homework assignments to you guys, and I may have another one due next Friday here pretty soon. Well, from the last homework, I wanted to mention a couple little things. Is it sharing? I think it's sharing. Okay, it's sharing. One, I made an error that I just wanted to bring up in the synthesis of this molecule, and it was going from the ester to the carboxylic acid, and the reagent is sodium hydroxide, NaOH, not sodium ethoxide. Of course, sodium ethoxide wouldn't really work because it'd be kind of going from an ethyl to another ethyl, right? You have an ethyl and add another ethyl. So you actually need a basic reagent like hydroxide to do the cleavage. The other thing is there's 2 tautomeric forms of this molecule, and they both equilibrate, so they're both probably present, and drawing one or the other is totally fine. When you consider the next reaction, which is reaction with a base followed by ethyl iodide, you could use either of these forms. So if you use the first form, you would imagine the base take off the proton, right? And then the N- would attack the carbon, kick off the iodide, and that would give the final product. But how could you do it with the other one? You could also imagine that the base would take off that proton and make it give you an O-, right? And then the O- could kick in, the double bonds could shift around, and then the N- could go attack. So using either of these tautomeric forms gets you to the right place. And so cool, that's that. Another couple things from this homework that I thought were cool. This is a cool paper, I thought. It was a nice, pretty readable paper. I'm sure you guys can agree with that. It was, some of the biology is a little crazy, but for the most part, it's a typical one of these JMedChem papers, Journal of Medicinal Chemistry. Some biology, some chemistry, and some potency, IC50 values and stuff. And so JMedChem is a pretty cool journal for kind of keeping up on the medicinal chemistry world. Let's see, one thing was that, of course, these are all the molecules they made. And as you guys know, and as we mentioned, what does plus minus mean? Like for 13D, this is the best compound. 13D, the potency is below, but the plus minus means it's, you know, something about the chiral center, right? There's a chiral center in the molecule, and it's a racemic mixture. So it's a plus. Plus would rotate light one way, minus would rotate light the other way. But by saying plus minus, you kind of assume a racemic mixture of both of the enantiomers, right? Okay, so we look at the potency, of course, and it's nice that 13D was the best compound. In terms of inhibiting the wild type HIV reverse transcriptase, and a couple of the mutant forms, it was fairly potent. But here's the question I have. Is there a way to make 13D more potent? Like that's kind of weird, right? Like 13D, that molecule, could we maybe turn the potency into, like, I don't know, about half of that, like 0.1516 micromolar. That's actually, these are micromolar, so it's 36 nanomolar, right? It's 36 nanomolar. It's 36 nanomolar IC50. Is it possible that it's actually more potent? That's what I'm trying to say. Any thoughts on that? Like, I mean, there could be experimental error, but I'm trying to argue that it is possible that 13D is actually even more potent, almost like double the potency, which would be about half the IC50. How could that be? Any thoughts? How could the IC50 actually be half of that? They're saying 36, but I'd say, well, no, it might actually be about 0.018, right? Half. Why half? What? Trying to push you guys in the right direction by talking about chirality, right? So what could possibly make it actually a more potent compound? Well, of course, if it's a mixture of enantiomers, that could mean that one of the enantiomers is twice as potent and the other enantiomer is totally dead, right? That makes sense? So it could actually be that the IC50 is about 18 micromolar, sorry, nanomolar, 18 nanomolar, right? 0.018 nanomolars, right? So how could we investigate that if we really wanted to? And you always have this issue with racemic drugs, right? What would be a potential way of maybe investigating this and saying, well, maybe produce the single enantiomers? Because the structure of 13 is roughly this, right? So it's this kind of molecule. So there's basically two options. Anybody have an idea? If our goal is to make the R version of 13D and then the S version of 13D, what are the ways? So there's you can do it through synthesis, right? We talked about a couple drugs where we could make the single enantiomers, right? But what if you're just a horrible synthetic chemist and you have a gram of this, a gram of 13D sitting around? How else could you do it? Say we have a gram of 13D that's racemic. Any ways to make the individual enantiomers? Or are you just stuck with it? Is a racemic mixture stuck as racemic forever? And you know possibly the technique, because you can't actually separate. It is possible to separate enantiomers, right? Anybody know the name of that technique? What's the name of the technique to split up or like resolve, we'll say, two different enantiomers? Anybody know? So I'll just click here. Yeah, the technique to resolve two enantiomers is called resolution. All right, so you can resolve mixtures of enantiomers. Now, there's a lot of ways you can do it. One of the ways is basically you can use HPLC, right? HPLC is an analysis technique, but you can use a chiral column in an HPLC. So, right, chiral HPLC is one way. Pretty cool. You just use normal HPLC with a chiral column. And the chiral column the column, of course, is how the separation takes place in HPLC. And if you have a chiral column, then one enantiomer will come through the column a little quicker, the other enantiomer will go through the column a little slower. And then you basically get two peaks out, one for the R, one for the S, which is a pretty super cool technique if you have a racemic molecule, right? Does anybody know, you may have heard about this in like organic lab course or something. Any other ways to do a resolution, to resolve two things? Because chiral HPLC costs a lot of money. You got to that column, this column, the chiral HPLC column is maybe $1,500, you know, maybe $1,000 or more dollars. And so chiral HPLC is great, but it's a little pricey. Any other ways we could separate a mixture of enantiomers? Classic ways are like crystallization, but you might need like some kind of chiral acid or something that will help distinguish the two enantiomers. Anyway, there's a big technique called resolution that discusses this. Okay, that's that. But I do have, like, one of my own research results uses this kind of idea, and I just want to show that briefly. So this is a paper I contributed to. Let me find it. So this is a few years ago, about 6 years ago, journal called Molecular Pharmacology, volume 88, page 689 to 696. It's also in the recording if you want to check it out. Sorry. Okay, so this is a complicated paper, but the main idea was we're inhibiting a protein called CFTR, which is a cystic fibrosis transmembrane regulator. It's the protein that is kind of responsible for cystic fibrosis. Okay, a lot of story behind that. And we were looking at a drug called BPO-27, which is created by my colleagues at UCSF. So we discovered this molecule. We did some synthesis, optimization of it, and let's see, the molecule is right here, BPO-27, R form. So the basic story was it has a chiral center, right? So we published the original mixture as a racemic mixture, right? And then we separated the mixture using chiral HPLC, and we got a mixture of R and S. And basically, then we were able to characterize the IC50 of the R and the S, right? Which was pretty cool. And then our potency doubled, right? That's exactly what I was just talking about. And then I'm kind of a amateur computational chemist on the side, and so I was able to do modeling of how the molecule binds to CFTR. We found a couple different binding sites. So like we didn't know which binding site occurred, so we bound this molecule to binding site A, B, C, and D, and E. And then I basically okay, so assuming that R is good, right? If R is the good one, and we determined that by we determined that by experiment. We did the assay, right? Assuming R is the good one, and I'm trying to evaluate these different binding sites, like, because they can bind to all these different binding sites, right? Assuming we know R is a good one, then how could we figure out which binding site's the correct one? We know R is good and we know S is bad, right? So what we did experimentally and using computers is I was able to bind R and S, 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 and I'd find the one that had the most complementary interaction where R seemed to bind the best, right? And so I did that as an experiment, and I found, I think it was D or E was the best one. And then we did like more computational stuff, molecular dynamics, which is a kind of lets all the protein equilibrate with the molecule. Not going to go through the whole paper, but in the end, we basically have a binding site for R. This is the R, which is also the ATP binding site. So it actually competes with ATP. This is ATP in the bottom and C. And R, the R form of this molecule, actually makes some of the same contacts as ATP does to some of these amino acids on the right. So it's like this R form of this molecule was an ATP mimic, essentially. And then the S form did not bind very well. And it had, it makes sense because the chirality is opposite, right? It had actually, the protein had to completely adjust itself to fit in S. So yeah, it was pretty cool. The other thing, let me see if I have it up here. It should be highlighted. And yeah, you're welcome to go check out this paper if you want. It's a little bit of a complicated sort of physiology paper, but check out the IC50 of R. The IC50 of the R form of BPO27 was 0.53 nanomolar. 0.53 nanomolar. So that's what? That's sub-nanomolar. So it's under a nanomolar. That's insanely potent, right? It just shows how intensely, how much of an intense affinity this molecule has with CFTR. And the R form, the S form has some affinity, but it's much less. And I don't think it's in this paper, but it's in the other paper that we wrote on the S form. But that's like picomolar. That's not, sub-nanomolar is picomolar, and that is incredibly potent. Okay, is that it for this? I think that's it for this. But yeah, pretty cool paper. I was happy to kind of contribute the computational modeling of the R and the S and the ATP, and basically look at all the different interactions. Okay, cool. Somebody said, can it convert into diastereomers? Can it convert into diastereomers? Well, certainly, if we look at this molecule, there's no diastereomers because there's only one chiral center, and you need like 2 chiral centers to be diastereomers. So yeah, that doesn't really make sense. And also that last molecule, the reverse transcriptase inhibitor, that's also a single chiral center. So there's no diastereomers. Okay, all right. So we're cool with that. Yeah. Now we're actually entering PowerPoint. A little bit of PowerPoint. Okay. Okay, we're still sharing, yeah? I think it should still be sharing. Okay, so we did some whiteboard stuff or whatever, pseudo whiteboard stuff. Now we're going to kind of continue a little bit in PowerPoint, all right? Okay, so one of the things before I do that is I just wanted to mention this thing called P-glycoprotein. It's called the efflux pump or the multi-drug resistance pump. That sounds kind of scary if you're trying to create drugs, right? Like a pump that's resistant to all sorts of drugs. And what it does is it essentially pumps drugs out of organs. And kind of maybe our body it's a defense mechanism because it thinks the drugs are bad and it wants to kick them out. And actually, and this happens in all sorts of types of organs, the liver, the small intestine and colon, tumor cells. This is really scary because if you're trying to treat a tumor, the drug can actually get, anti-tumor drugs can get kicked out of tumors. The testes, the brain, the kidney, even the uterine wall, all have this P-glycoprotein in it. And that was the other kind of interesting thing we mentioned was Loperamide, which was the anti-diarrhea drug. It was an opioid, right? And this one actually, so the reason that it's a nice over-the-counter opioid that works well as an anti-diarrhea drug is even though it does cross the blood-brain barrier, but it gets kicked out basically from the inside of the brain out to the the blood vessels, and then it gets kind of lost. So it doesn't actually have a psychological or effect on your brain like the way like fentanyl morphine and things like that. So like an analgesic effect. So yeah, it works really well as an anti-diarrhea drug because it only kind of works on smooth muscles, right? That's kind of cool. This was a X-ray crystal structure I found of P-glycoprotein. It's a channel protein. This is from C. elegans, which is a roundworm. Kind of an interesting protein, like a lot. Oh, that's basically where it was isolated from for the crystal structure. But it's a pretty similar structure to like humans and mammals. And it's basically it's a channel protein that sort of just kind of binds to certain molecules that have certain structural features, maybe slightly polar molecules, and it kicks them out. It kicks them out from the inside of the cell to the outside of the cell. Okay. Yeah, you can try. You could actually the PDB code is 4F, 4C. You could try it in IC3MD if you like, or if you're interested in protein crystal structures, if you get Pymol working, you could actually use Pymol. Generally, all of my crystal structures in this presentation are all Pymol, just because I love Pymol so much. Okay, good. All right, so what did we talk about on the whiteboard? We did some definitions. We talked about basic pharmacodynamics and basic pharmacokinetics, a little bit on clinical pharmacology. We talked about toxicology a bit, which I think is a really cool topic, especially since it's complementary to pharmacology. And it's very interesting to know how drugs are toxic or molecules are toxic. And also it may help you, like when you're working in the lab, to make sure you avoid exposure to toxic chemicals. Okay, so what are we going to discuss next in this series of slides? Drug names and nomenclature, which is not that exciting, but I just want to bring it up a little bit. And this is following that the pharmacology wiki file that's on the iLearn site too. Some discussion on drug targets. So a couple types of drug targets. Drug receptor interactions, so the kinds of interactions that drug binds to a protein or something, and how does it bind and interact, right? That's a good question. A little bit of dose response, which we've seen a few times now. Then the concept of agonists and antagonists. Okay, all right. Okay, so anybody know what this molecule is? If you teach, I don't know if any of you teach Organic 2 Lab, it's experiment 1. So this is a very simple molecule. Of course, this is aspirin. So a question is, how do we name it? Well, naming molecules is a little complicated. There's a couple ways, even in chemistry, to name things. IUPAC is a kind of a most systematic naming system. One of the names that this could have is acetoxybenzoic acid, where benzoic acid, of course, is the base. carboxylic acid, and then 2-acetoxy refers to this oxygen and ester being at the 2 position. There's another name. There's something called common naming, which you will see as you the more chemistry you do and the more chemicals you encounter. And this is actually a pretty common name that people use when they describe aspirin. They call it acetyl salicylic acid. Acetyl salicylic acid. So that suggests it's an acetyl group on salicylic acid. And salicylic acid is this molecule, right? And so actually it's also the synthetic precursor. So if you're making aspirin, you usually just take this stuff and you throw in an acetylating group like acetic anhydride, and then that converts this into this. And that's what we do in Organic 2 Lab. The first experiment is we make aspirin, right? Okay, so generic name, aspirin is every molecule has a kind of a non-proprietary pharmaceutical name. And then there's like trade names like Bufferin, Ecotrin, Emperin that are kind of used by manufacturers and they, in their marketing campaigns and stuff. Okay, proprietary names. What about this molecule? All right, so this is Tylenol. As a chemical name, and I mean, this is not totally logical. I could probably come up with 10 different names of this, but it's kind of saying acetamide. Acetamide is just, of course, this thing on the right. And then it's saying N4-hydroxyphenyl. So it's like this thing is on the acetamide, N4-hydroxyphenyl, acetamide. Another common name, which is not, both of these are totally acceptable. I would say IUPAC is a little more systematic, but N-acetyl para-aminophenol. This is kind of like called it's kind of a common name. Phenol, for example, refers to the benzene with the hydroxy group on it, and then N-acetyl would refer to the acetyl group being on the nitrogen. So not totally logical, but you do see this, and it's nice to kind of think about it a little bit. And then we have generic names, acetaminophen or paracetamol. You've probably heard of those before. And then the trade name would be Tylenol. Okay, so this is the anti-tumor drug 5-fluorouracil. So it's a pretty effective chemotherapeutic. Chemical and IUPAC is just 5-fluorouracil. And then the common name is actually the same thing. And the generic name is also the same thing. So these are a very common naming system for, some drugs are like this. And then it has like a trade name like Adrucil. Okay, so this is a case where a couple of the naming systems are the same. All right. Anybody know what this is? It's a classic, complicated molecular structure. The molecule is pretty incredible, and it's a very potent anti-cancer drug. My postdoc advisor at UCSF was the first person to make this at Scripps Institute. It's called Taxol, right? This is Paclitaxel, and a very complicated structure and a very, very, very challenging synthetic target. I mean, imagine how would you go about making this from very simple molecules, and the total synthesis is like 20-25 steps or something from very simple molecules, and every part of this is synthesized and all the chirality is correct. It's a complicated thing. That would be a fun thing for me to try to present, actually, the synthesis of this. But you kind of need to know advanced synthetic chemistry to really do this. This is not your Organic 2 level stuff, right? But yeah, this is Paclitaxel, is the non-proprietary name. And the trade name is Taxol. So this is used for all sorts of types of cancer, prostate cancer, I think colon cancer, and a couple other types of cancer. It's a injectable substance, so you take this as another chemotherapy, all right? Okay, what makes a molecule a drug? Any guesses? What makes a molecule a drug? What are some thoughts? That's kind of a general question, right? So it's a little hard to answer. You'd probably say something about, it has to have maybe a therapeutic effect, ingesting it through some manner. But should as opposed to a toxin, which would actually cause a toxic effect. One definition would be a chemical that perturbs a biological system for a therapeutic effect. That's a good definition, right? So what are drug targets? I would say most of the time, proteins, DNA, etc. Are there any other non-protein or DNA drug targets? I think I mentioned a couple of these. Anybody remember? So if we're not inhibiting like a protein or like binding to DNA, what else? What other targets could a drug interact with? RNA, yeah, that's an interesting question. RNA is somebody said RNA. RNA is kind of a hard one because RNA is kind of short-lived usually, and it's, are there any RNA targeting drugs? There probably are, but it's kind of a rare one because they are sort of like messenger RNA is usually kind of a short-lived thing, and also RNAs are kind of unstable due to RNases. Hormones, yeah, and that's an interesting idea, and there's definitely there's peptide hormones and things that drug might interact with. Yeah, those are a couple ideas. And then some of the ideas that also that I kind of found just reading around. Of course, there are simple things like stomach acid, right? If you have heartburn, you could take an antacid drug like calcium carbonate, and that would actually kind of neutralize some of the acid in your stomach, and that's actually a nice approach. We also talked about like Ametrazole, which was another antacid type drug, but it actually targeted the proton pump in the stomach. So that was actually a protein targeting drug. Other things, this is an interesting one, like plantar warts. I don't know if anybody's ever had a plantar wart. They are really painful. I got one maybe 15 years ago from walking around in the gym without slippers on for a bit, and very painful. And anyways, a lot of the treatment strategy is really to burn it off. And this is just a super acidic molecule. So this is almost like a toxic substance that you can localize onto the surface of a wart. So there's different brands of this, but it's a really simple molecule. It's just chloroacetic acid. Is that more acidic or less acidic than acetic acid? Acetic acid, of course, is just that. And anybody remember why? That's an interesting question. You might make a guess that the chlorine probably affects the acidity. And maybe if it's killing a wart, it's probably going to be more acidic than vinegar, right? And it is. pKa is 2.8, which is still not it's not like sulfuric acid, right? That's not as acidic as sulfuric acid, which is like in pKa like -4 or something like that. But it is more acidic than vinegar, which is 4.7. And that little bit of extra acidity is what you need to effectively kill a wart, right? Remembering also that pKa is exponential. So in terms of the proton coming off the acid, you might say, oh, that's not that big of a difference, right? But it's 10 to the difference. The difference is almost 2, right? 10 to the 2 is 100. So this is actually 100 times more acidic, and that actually has a nice efficacious, effect for killing warts. Now, there's if you put a fluorine on, it's even more acidic, or like 3 fluorines, trifluoroacetic acid, is even more acidic. But then you start getting kind of non-specific effects and burning your skin and things like that. So this happens to be just like an optimal kind of strong acid for wart treatment. Protein receptors or DNA. So a couple types of protein receptors that we'll mention. One is called a ligand-gated ion channel, a ligand-gated ion channel. What would that mean, a ligand-gated ion channel? I mean, it kind of sounds like a drug might bind to the protein and then open up a channel. That's exactly what it is. And then ions will either enter the cell or leave the cell, and they'll have some kind of change of ion concentration in the cell that'll have a pharmacological effect. So that's kind of one class of drugs. Another one are called G protein coupled receptors. So usually what this does is a drug will bind on the outside of the cell to a protein, and through a kind of a cascade process, proteins get phosphorylated on the inside of the cell, and that has some kind of cell effect on the cell. Now, these drugs are not making it into the cell, which is nice because it's kind of hard for drugs to get through the cell, right? Because the membrane is often impermeable. Enzyme-linked receptors. So in this case, a protein binds to the receptor, and that causes like activation of the protein as an enzyme on the inside of the cell. So that might phosphorylate something and cause some kind of downstream effect, okay? So enzyme-linked receptors is another type of drug target. And then intracellular receptors, these are kind of cool. So some steroids, what they do is they sneak through the membrane because they're greasy, and then they can get inside and they'll bind to a receptor on the inside of the cell and then cause some phosphorylation or something like that. So there's a couple types of things. And then of course we also have like enzymes that are out on the outside or on the inside of the cell that the molecule can bind to also, okay? DNA or RNA, I guess RNAi therapeutics are an interesting other type of thing, and that's like, RNA that will inhibit gene expression. So it's a type of RNA that's actually used as therapy, I guess. I'm going to look for uses of RNA as an actual drug target. I can probably find an example of that. Okay, so drugs that bind to DNA might kind of sneak into the base pairing of DNA, right? And that's kind of and that can actually have, some anti-cancer drugs do that. So they'll sneak in to the DNA and they kind of perturb the structure. Okay. Now this is a Thidium Bromide, which is kind of interacting with the alpha helix of DNA. And Ethydium bromide is not really a drug, but you do see it in the laboratory. It's used as it stains DNA gels. So if you do DNA electrophoresis, it's this kind of weird molecule. And of course, what kind of structural feature does this have that might allow it to sneak into the base pairs? Anybody see anything that kind of looks interesting? Knowing that DNA is kind of held together by like pi stacking of the bases, right? So what's this have that might interact with DNA base pairs? Somebody said nitrogen bases. It kind of looks like a nitrogen base almost. You have these anilines, but it's maybe the best way to think about it is kind of an extended aromatic system, right? Like these fused aromatic rings. Those are flat and planar and can just sneaks into the DNA base pairing, right? Now, are there actual drugs that do this? Yes, there are. This is a nice example called D-Actinomycin, or the drug name, the trade name is actually D-Actinomycin, so that's pretty clever. It's a chemotherapeutic DNA intercalator that's used in cancer. A lot of these things will basically just inhibit cancer cell replication by, perturbing DNA structure. Okay, and we'll see. I think I have a figure of this one a little bit later, of how it actually fits into the DNA structure. Okay, let's see a couple of these drug targets by X-ray crystallography. Okay, so what was the ligand-gated ion channel again? So that was kind of a ligand binding to a protein that's a channel, and then it allows ions to flow either one way or both ways or whatever. So a good example is the nicotinic cholinergic receptor in complex with nicotine. Okay, so this is the protein. These have really cool structures. It's a huge multi-protein complex. Nicotine is the thing up here, the little red spheres. What happens is it's a pentamer, so sodium and potassium will conduct through the middle of it, so it's an ion channel, right? So nicotine is that molecule, and when it binds, it opens up and allows sodium to flow into the cell and potassium to flow out of the cell, and that has kind of a lot of the effects that nicotine has as a mild stimulant. Other agonists, so nicotine is an agonist of this. Other agonists are acetylcholine, which is a kind of pretty simple molecule, and it turns out that acetylcholine. So there's a couple of these cholinergic receptors that bind to acetylcholine as a natural ligand, and there's a nicotinic one and a muscarinic one. They're like subtypes, but they bind to acetylcholine also. Epibatidine causes numbness and paralysis and possible deaths. It's kind of like it's a poison, basically. It's found in the poison arrow frog. So we have a few of these frog species that have poison chemicals inside them with really cool structures. I love these little bridged kind of weird molecules. Okay, so 6PV7 and PDB, if you want to check it out. G protein coupled receptor. So the classic example of this is the beta adrenergic receptor, which is adrenaline receptor, which causes kind of like hypertension and increased heart rate and blood pressure and things like that. And it's a target for drugs that are, block hypertension, so antihypertensive drugs. And one of those drugs is called carazolol. Okay, so yeah, it's a pretty simple protein, relatively speaking. And so then the molecule, this is an antagonist, blocks or binds to the receptor and basically inhibits downstream function and phosphorylation and things like that from within the cell. So GPCRs are a very common class of drug targets. There's a lot of these, and they have agonists and antagonists that cause some kind of pharmacological effect. So about 34% of all FDA approved drugs interact with GPCRs. And there's about 108 GPCRs that are kind of used for different diseases. So it's a very common class of drug targets. So then basically what happens is G protein phosphorylation happens inside the cell. That leads to increased heart rate, blood pressure, etc. So if you want to block, increased heart rate and blood pressure, you can maybe design a molecule that will act as an antagonist of this receptor. And that's exactly what carazolol does. It's a beta adrenergic antagonist. We call them beta blockers. There's a whole bunch of beta blockers. We talked about beta blockers a little bit, I think, in the synthesis stuff. They often have an alcohol in them, and that was a chiral one. I don't know if this is actually a racemic drug. Probably, I think most of the beta blockers actually have a single enantiomer of an alcohol of them. But yeah, it's a nice, effective way to reduce blood pressure and heart rate. And that has the downstream effect of kind of blocking these sort of downstream things, okay? Indinavir, HIV-1 protease inhibitor that we talked about at some point, a little bit. What do these do? The protease inhibitors, they, here's a great little summary, of course. So you have kind of viral mRNA, and those get translated into a big polypeptide, polyprotein type structure. And then the proteases, HIV proteases, will kind of break apart that big polypeptide, polyprotein, and make the different components of HIV: matrix, capsid, nucleocapsid, reverse transcriptase, more protease, transmembrane proteins, et cetera. All of these proteins are needed for HIV to be HIV. And COVID kind of does something like this too. Kind of does something like this also. But anyway, so this is a great, cool example of a protease inhibitor bound to the protein, and this is it's a dimeric protein, and also it's called an aspartic protease because there's a couple of aspartic acids, that do the cleavage. So you can consider a protease like a pair of scissors, right? A protease is a pair of scissors, right? And the scissors in HIV protease are controlled by these aspartic acids, and the aspartic acids protonate peptides, and that causes the easy cleavage of them. And what do I have the structure of this? Oh yeah, there it is. That's the structure of indinavir. It's one of the early HIV protease inhibitors. They got some better ones now. But aspartic acid is an amino acid, right? And it's aspartic acids, are they protonated at physiological pH? Aspartic acid is a carboxylic acid, right? What does it look like? I can't draw right now, but is it really a carboxylic acid? pKa of an acid is 4, right? Physiological pH is about 7.85 or something like that. So aspartic acid is actually deprotonated. It's a negative charge, right? It's a carboxylate. And the carboxylate, the negative charged carboxylate, it actually is binding to that alcohol. So the alcohol is a really critical part of this structure, right? And so you have this kind of scaffolding that's sort of like peptidomimetic. It almost kind of because what usually fits in this hole? A peptide, right? The HIV polypeptide. And so this has to kind of make some of the similar interactions that that the HIV polypeptide makes, and this is kind of pseudo, peptidomimetic. That's the word, peptidomimetic. All right, yeah. I think this is one of the first really cool examples of this concept of structure-based drug design. The idea of using X-ray crystallography in the drug design process. So they were able to discover, the structure of this protease, and then kind of using iterative drug design and kind of, making analogs of this molecule, they're able to make something that actually was efficacious. It blocked the protease. It was stable enough that it won't just be degraded by like liver enzymes and things like that. And it had an affinity that was able to basically bind into this protease and block it, and not block like human proteases. So it's very selective. I would say, yeah, one of the cool things about HIV in terms of drug development was it let us practice some really cool techniques like structure-based drug design. Okay, this is another alternate image that kind of shows the surface of the two proteins, and this molecule kind of snuck in there, and it's just a blob with a hole in it, and the hole is where the proteolytic activity takes place. Okay. Zidovudine, you guys are familiar with. It's the HIV reverse transcriptase inhibitor. I think I may have shown this structure before, but this is how it's covalently attached to a growing strand of viral DNA. So when HIV reverse transcriptase is converting RNA to DNA, zidovudine or AZT kind of gets stuck in there, and you have the three nitrogens of the azide that terminate its growth, right? Cool. And we also had, what was that? Maybe it was, I think it was this last homework assignment, homework 2. There's also those things called non-nucleotide reverse transcriptase inhibitors, right? Non-nucleotide reverse transcriptase inhibitors. Did anybody catch, what is the orange loop? Oh, that's somebody asked, what's the orange loop? Well, that's the DNA. That's HIV DNA, right? It is basically converting the RNA strand into a DNA strand, and the DNA strand is growing, and then AZT gets incorporated, and that kind of blocks the growth. So the azide blocks it from continuing. And then remember those non-nucleotide reverse transcriptase inhibitors. Well, those bind to a different part of the protein, right? Because they don't look like nucleotides or like AZT. So they bind like to an allosteric site or something else, elsewhere on the protein that kind of prevents this from operating, which is actually pretty cool that you can do that, right? So this is a big protein, and it has multiple potential binding sites, but AZT specifically and covalently attaches to the growing DNA and blocks it. So there it is, AZT, zidovudine. This is just another vantage point, and it just shows that this HIV protein is actually a pretty big, moderately complicated protein. Okay. Here's our D-Actinomycin or D-Actinomycin. It's a DNA intercalating chemotherapeutic drug. And it's going to show that idea of the intercalation, right? It intercalates into the DNA base pairs. So let's see. This is a. Let's see. There's the protein. I mean, the molecule is kind of sneaking into the base pairing of the DNA, right? This shows atomic structure, and it kind of shows where all the bases are. You have all the bases of the DNA strand, and this is a big, complicated molecule with lots of fingers, and all the fingers kind of interact with the DNA strand, and that perturbs the structure. And again, it's used as a like an anti-cancer drug. So it's an injectable mixture that can be used in certain types of tumor inhibition. Okay. DNA intercalating is the word. Okay, so drug receptor interactions. What are some of these drug receptor interactions? You may have seen this before. I don't know. It's I mean, a lot of this is kind of just molecular interactions that you've learned about in Gen Chem and places. So what are some types of interactions? Anybody know any? Like what are the processes, just basic chemical processes where a drug can bind to something, like a protein? Simplest ones, intramolecular interactions, hydrogen bonding, that's a great one, yeah. And there's other things kind of like that, like electrostatic interactions, dipole-dipole interactions, and things like that. But yeah, hydrogen bonding is a great example. Hydrogen bonding, or also van der Waals, dipole-dipole interactions. So this is how atorvastatin kind of interacts with HMG-CoA reductase, which we've talked about earlier. It's it's an enzyme involved in cholesterol biosynthesis, right? And atorvastatin, Lipitor, is a cholesterol lowering medication. So it's got a lot of like protons, and it's got the carboxylate. This has alcohols, right? It has alcohols, and the carboxylate and the hydroxy groups, they can all interact with either protons or like other carboxylates in the enzyme. So all of these are hydrogen bonding interactions. So that's Lipitor. In terms of the van der Waals or dipole-dipole interactions, an example is this antidepressant drug, pivagabine. Some of the pronunciation I'm probably gonna get wrong, but it's an interaction between like a negatively charged thing and a positively charged thing. And sometimes there'll be a combination of electrostatic interactions and also hydrogen bonding. So the negative charged oxygens could potentially bind to the NHs as well. This is a nice example of a dipole-dipole interaction. Zeleplon is a, what is this? I forgot what kind of drug it is. Oh, it's a sedative hypnotic. Okay. But anyway, look at some of these cool interactions, like a hydroxy group, a delta negative hydroxy group binding to the nitrile, which is delta positive. So those kind of interactions are electrostatic, right? Something negative with something positive. A carboxylate binding to like delta positive amide, etc. So these are, it's like this is a dipole-dipole interaction, right? You have two dipoles that are interacting. This is called an ion dipole interaction. So you have an ion, like a negative charged ion, interacting with a dipole, which is delta positive. Hydrophobic interactions. So like kind of greasy drugs can interact with greasy receptors. A greasy drug structure and a greasy receptor structure can interact. And so one of the mechanisms, this is proposed a long time ago, 1970. So the idea would be that if you have like this is actually from the original book, like a receptor greasy chain, like maybe a leucine or isoleucine or something like that. So there's a kind of an entropic model for this. The idea is you have, if you have a greasy thing, there will be a lot of water around to kind of stabilize it. It's kind of, and it'll require a lot of solvation. The drug will also have this kind of effect. And when the two things come together, now that frees up more of the water to be released, and that's kind of like an entropic effect. So the more water is kind of released, it's not right. This is very ordered, right? On the left, it's very, very ordered, so it's not very much entropy. But on the left, there's more entropy because water is kind of released to the pool of solution. So this is a model for hydrophobic drug interactions proposed in 1970. Essentials of molecular pharmacology, a long time ago. Example, butamabine is a molecule with a butyl ester and then like an isoleucine. This is a local anesthetic, like a cream. Okay, so these kind of interactions, greasy drugs can have these sort of interactions with a drug target. What else do we got? Covalent, right? We talked about this a bit. Remember suicide substrates? The idea that a molecule can be permanently attached to an amino acid. Where we introduced that was clavulanic acid, which was the, what was that again? It was a beta-lactamase inhibitor, right? It says, right, the antibiotics like the penicillins and cephalosporins would get cleaved by beta-lactamase, and this would block the cleavage process. So that's clavulanic acid. It's a beta-lactamase itself, a beta-lactam itself. And what actually happened, remember, was serine 70 would actually go attack this. It would attack the beta-lactam, open it up, but then be permanently attached. So serine 70 is kind of permanently turned off, I guess, or it's permanently deactivated, right? Charge transfer interactions, this is another type of interaction. It's kind of cool. A complex between an electron-rich and electron-poor aromatic ring system. An electron-rich and electron-poor. What does that mean? Some aromatic rings have a lot of electron density, and some have kind of a lack of electron density. And those things can kind of interact. Isn't that weird? I mean, it makes sense. It's called a charge transfer interaction. So as an example, chloranthrolin, what is this molecule? It's actually a fungicide. It's a fungicide. So it has, imagine, let's get rid of this stuff. What do the chlorines do to that benzene on the left? The chlorines, are they electronegative or electropositive? And also the nitrile. The nitrile is electronegative, electropositive. Are all these things in the benzene, are they delta negative or are they delta positive? So definitely very negative, right? They're delta negative. And what does that do to the benzene? It makes the benzene delta positive, right? So the benzene is kind of, we say the benzene on the left is electron deficient. But a tyrosine amino acid, it's got an oxygen on it that kicks electron density in, right? And that makes the right side benzene electron rich. So left one's electron deficient, right one's electron rich. And that actually forms a interaction, okay? Okay, so anyway, yeah, this molecule is a fungicide, wood protectant. It's a suspected carcinogen, not really used as a drug, but it's a cool example of this type of interaction. It's this stuff called Bravo, which is, I think, is probably like lawsuits on this stuff. So don't take this as a drug. Let's talk about how, oh yeah, another one, pi stacking, pi stacking. So this is the idea of, it's kind of similar to a charge transfer complex, but it's really just like two pi systems that are close and not necessarily, you know, one's donor, one's acceptor, like we just drew, what we talked about. So like a benzene and another benzene, that's a pi stacking interaction. Lacosamide is a anticonvulsant, anti-seizure drug, anti-epilepsy drug. So these kind of interactions is not a tremendously strong interaction, but you'll have maybe other interactions on the molecule as well, okay? Pi stacking, of course, is a critical component of DNA alpha helix structure that we talked about. And we saw that with like, yeah, D-Actinomycin. So this just kind of shows, you know, DNA structure, of course, there's hydrogen bonding in the base pairing, but there's also this kind of pi stacking. And the intercalators will try to sneak in there into the pi stacking, and kind of, it's crazy that that could be used as a drug, but those are good therapeutic approaches. Another type of interaction, cation pi, a cation pi interaction. What do you think that means? Cation pi. Well, a cation is something positively charged, and a pi would usually be like a benzene or something. And why is that an interaction? That's kind of crazy. It is, and there's a bunch of examples of that. And here's just an example, kind of illustrating it. This molecule, this dexamphetamine, which I think is an amphetamine derivative, but it basically has this NH3 plus, right, as a cation, and that can interact with benzenes and tryptophans, indols, things like that in the protein structure. And these are very common in lots of types of drug interactions. And so, yeah, this is a stimulant that's used as a ADHD drug. Okay. Cation pi interactions are found in a couple different things. Drug protein interactions, right? So drugs and proteins, but they can also be like protein structure. So like a protein will have these cation pi interactions as part of its internal structure, and that'll stabilize the structure, basically, right? And yeah, so that's cool. I think I'm going to stop here, and I'm going to keep going with the asynchronous lecture. And yeah, I think that now's a good time to stop. But what I'm going to do is kind of keep going with this. I'm going to show some real examples of this cation pi stuff. All right. I think I'll do that. All right. Any questions? I think it's cool to see these in real crystal structures, and seeing the protein structures of drug targets. I'm always fascinated by that. Can I check in with you after class? Yeah. Somebody was asking me that. Yeah. Okay. I'll stop recording.